Welcome to day 9. Today I'll be presenting on CRISPR technology, which is a technology that has revolutionized how we think about genetics and medical treatment and even human evolution. And this is a really powerful technology that can make us naturally immune to almost all diseases. And it's one of the most powerful gene editing tools ever discovered by humans. So, what is CRISPR? CRISPR sounds really complicated right here. The long name, clustered, regularly interspaced short palindromic repeats. It just describes a repeating pattern found in bacterial DNA. The easiest way, this is really hard. It was complicating for me too to interpret. The easiest way to think about CRISPR is as a DNA archive or a kind of genetic bookshelf. So basically, when bacteria survive a attack by virus, they store little pieces of the viral DNA on this shelf known as CRISPR. So the next time the same virus attacks, the bacteria can recognize it instantly and kill it. It's kind of like similar to a vaccine system. Um, it's similar to how immune system remembers virus, but it's just DNA. This natural mechanism is what scientists realized they could reprogram for gene editing. CRISPR technology, it doesn't work by itself. It needs a partner. Well, it needs a partner which does the main role in this whole process, the Cas9 enzyme. Cas9 is nicknamed the genetic scissors because it can cut DNA extremely precisely. Cas9's original job in bacteria is to cut apart viral DNA once the bacteria recognize the virus from their CRISPR memory. But scientists realized that if we reprogram Cas9 with a guide RNA, often known as gRNA, we can instruct it to cut any DNA sequence we want. Like if we want a specific DNA strand to be modified, you can program the gRNA to locate that strand and cut it using Cas9 enzyme. One important thing to note is that Cas9 doesn't not work alone. It does nothing unless it receives instructions from the guide RNA and finds the correct signal called the PAM. And we'll learn that in after 2 slides. Once Cas9 cuts the DNA, the gene at that site becomes inactivated. So scientists can insert a new gene into that location or just remove it. So Cas9 leads the entire shutdown or editing process, and that makes it the most crucial part of CRISPR technology. So right here, if you see um, the blue RNA DNA strands are being cut by the Cas9 enzyme, and a new kind of DNA is going to be inserted. Look, described by this purple DNA strand. And this is basically how CRISPR works. Now let's talk about something a lot of people overlook, the PAM sequence. The PAM stands for product spacer adjacent motif, and you can think this as a road sign that tells Cas9 you can cut here. Cas9 will not cut anywhere unless a PAM signal is right next to the target DNA sequence. So next to the PAM, there's the target DNA sequence that the Cas9 has to cut. This is extremely important because if the PAM sequence is not present, CRISPR simply cannot edit that location. So basically, Cas9 cannot edit that location even if the rest of the sequence matches. Because of this, scientists must design the guide RNA that can target a sequence with a PAM nearby. And in a way, PAM acts like a permission slip or a green light to for the Cas9 to start working. So basically, the guide RNA has to be reprogrammed in order to match the PAM2. And right here, the PAM is the yellow thing right here. It is right next to the target gene that has been modified, and the Cas9 just cuts it off. So now I'll walk through the full CRISPR editing process in 5 steps. Uh, More specifically, Step one is the targeting the PAM sequence. Cas9, guided by this gRNA, first searches the entire genome for a PAM signal. And also gRNA, and there's also a guiding RNA called the CRRNA. And those two basically guide the Cas9 enzyme to the target DNA they want to edit. This step is like scanning pages in a book for a specific bookmark, and the bookmark is the PAM signal. Step 2, matching the DNA sequence. Once Cas9 finds the PAM, it checks the DNA next to it to see if it matches the guide RNA we designed. If the sequence doesn't match, Cas9 moves on. If it does match, we move to step 3. And in step 3, Cas9 binds tightly to the DNA and begins to unwind the double-stranded structure, preparing to cut it. And then Cas9 cuts both strands of the DNA exactly at the target site. And this is where the gene becomes inactivated. After the cut, the cell naturally attempts to repair its DNA, and scientists take advantage of this repair process by inserting, deleting, or replacing specific genes. And this is the moment where the actual editing takes place. So CRISPR itself, reiterating, doesn't insert genes. The repair process does. The CRISPR process itself is just removing the gene that is targeted with extreme precision. And why is CRISPR technology important? CRISPR is considered one of the most promising advancements because it offers solutions to genetic diseases that currently have no cure. Unlike traditional medicine that treats symptoms, CRISPR targets the root cause of that specific disease, the DNA itself. Diseases such as sickle cell anemia, certain cancers, inherited blindness, and etc. could be potentially be corrected permanently. So it can be permanently removed. No possibility of getting that disease again. Beyond treating diseases, CRISPR also plays a role in genetic research, agriculture, and evolutionary studies. And yeah, for example, scientists can prevent the inheritance of harmful genes, study how traits develop, and even protect endangered species. Yeah, this is not fully researched yet, but we're hoping to do that, and it's in research process. Yeah, so in simple terms, CRISPR is not just observing biology, but we're actively shaping it using CRISPR technology. So we're changing the whole biological system, the evolution, genetic technology, everything through CRISPR technology. By understanding the CRISPR archive, the Cas9 scissors, the PAM's signal, and the repair process, we can now edit DNA with a level of precision that was once considered impossible. And this technology has the potential to transform medicine, biology, and our the understanding of life itself in the world of biology. And that's it for today. Thank you for listening, and I'll see you on the next video.